A pleasant good morning to all. I'm Dr. Srujna Kiran, Department of Microbiology. So today's webinar topic is Microbial Biotechnology for Crop Improvement and Disease Resistance. It's an immense pleasure to introduce eminent speaker, today's resource person, Dr. Padala Thirupathi sir, from SRR Degree College, Degree and PG College from Karimnagar to you all. Dr. Padala Thirupathi sir is an assistant professor of botany with 21 years of teaching experience and 15 years of research expertise in plant taxonomy, ethnobotany, and environmental conservation. He holds a PhD in botany from Kakatiya University and multiple postgraduate degrees in education, psychology, and English. He has published over 50 research papers, presented at 171 national and international conferences, and supervised PhD and MPhil scholars. He has received UGC research grants and was honored with the Best Research Award. Dr. Padala Thirupathi sir has held key leadership roles, including NSS programs, officer, NAC coordinator, IQAC member, and convener of Telangana Ko Haritaharam and Eco Club. He is also a slate level master trainer for elections 2023-2024. With 25 plus FDPs, 40 plus workshops, and multiple MOOCs, he actively promotes academic excellence. His contribution to student mentorship, electoral literacy sweep coordinator, and environmental I will hand over the session to today's speaker, Dr. Padala Thirupathi sir. Sir, please continue the session, sir.
Okay, very good evening to all the dignitaries and all the faculty members, all the students of various colleges and also the residential degree college. So thank you very much, madam, for your nice introduction about me. And it is a great day. And also thank you very much. Madam, the slide is visible. My PPT is visible.
Yes, sir.
Okay. So very good evening to all the participants. So Telangana Social Welfare Residential Degree College for Women, Kottagudem, District Bhadradri Kottagudem, Department of Botany and Microbiology, collaboratively organizing this one-day national webinar on microbial biotechnology for crop improvement and disease resistance. So in this regard, I would like to thanks to the chief patron of this national webinar, Dr. V.S. Alugu Varshini Garu, IAS, Secretary, TGWREIS, and the patron, K. Sarada, Joint Secretary, Madam Garu, and Chairman, Srimati Alivelu Garu, and Chairperson, Dr. B. Jansirani Madam Garu, respected principal of this sacred institution. And also I would like to thanks to Chief Advisor, Dr. PSR Sharma Garu, and Advisor, U. Zwalita Garu, and convener of this one-day national webinar, Dr. K. Sumalata Madam Garu, Head Department of Botany, Telangana Social Welfare Residential Degree College for Women, Kottagudem, and also co-convener of this national webinar, Dr. G. Srujana Kiran Madam Garu, Department of Microbiology, and the organizing members, Dr. K. Swati Garu and Dr. K. Sweta Garu. So thank you very much for giving me this opportunity to present this one-day national webinar as a resource person to each and everyone. So this is Dr. Padala Thirupathi, Assistant Professor, Department of Botany, Food Science, Crop Production, Microbiology, SRR Government Arts and Science College, Autonomous, Karimnagar. Am I audible, madam? Is there any?
Sir?
Am I audible?
Yes, sir. Yes, sir.
Yes, sir. It is audible.
Okay. So today our topic is microbial biotechnology for crop improvement and disease resistance. Then microbial biotechnology, it is involved the use of microorganisms like bacteria, viruses, and fungal members to enhance crop growth, improve soil health, and develop resistance against plant diseases. It is a key component of sustainable agriculture and food security. The main objectives of this today's national webinar to enhance the soil fertility, the soil fertility by using these microorganisms, and also developing sir webinar lo unnanu sir, developing biocontrol agents and improving plant growth and yield, and engineering disease resistance crops, enhancing abiotic stress tolerance. These are the main objectives of this national webinar. Then what is the definition of this microbial biotechnology? It is the application of the microorganisms like bacteria, fungal members, and viruses, including the microalgae, to improve agricultural productivity, enhance soil fertility, control plant diseases, and produce bio-based products. The microbial biotechnology also plays a crucial role in sustainable agriculture and environmental protection. It is a major problem we are facing present scenario in the global warming, climate change effects, and also the natural calamities we are observed in not only in our India but also in global perspectives. Then the key microorganisms in crop improvements, the rhizobacteria. The rhizobacteria, including Rhizobium leguminosarum, Rhizobium species, and also Azospirillum. These are the well-known bacteria for the fixation of the nitrogen. So please, all of you, don't write on the screen. Please listen. Raghavan. Who is Raghavan? Okay. The rhizobacteria, including rhizobium species and azospirillum bacteria, well-known bacteria for the fixation of the nitrogen from atmosphere. And some of the mycorrhizal fungal members. The mycorrhizal fungal members also improve the nutrients in the soil and also the absorption of the nutrients from insoluble to soluble nature. Trichoderma viridae and trichoderma also a fungal member which is useful for the disease suppression. Some of the actinomycetes microorganisms producing antimicrobial compounds. These are the useful for the crop improvements. Then microbes in disease resistance. Along with the crop improvement, microbes also used for disease resistance. Produce antibiotics and antimicrobial compounds and compete with plant pathogens for space and nutrients, induce systemic resistance in plants, enhance plant immune response against diseases. Okay. Am I audible? And what is the role of microorganisms in crop improvement? Plant growth promoting rhizobia or rhizobacteria, we can also call it as PGPR, are very useful in the beneficial activities that colonize plant roots, enhance plant growth by various mechanisms like nutrients solubilization, the phytohormone production, and suppression of plant pathogens. We know that nutrient solubilization means the nitrogen, phosphorus, sulfur. These are the mineral nutrients which are available in the soil, but it is insoluble condition in soil. By using of this rhizobacteria from insoluble to soluble nature, phytohormones also very important for the involvement of the growth and development of the plants, and also increase yield, crop improvement also. And some of the fungal, bacterial and other microorganisms also infect the plant roots and also the plants. So these microorganisms helpful for the suppression of the other plant pathogens. Then mechanism of PGPR action. So plant growth promoting rhizobia bacteria, how to involvement of this, the control of the pathogen and also the increase the development of the plant growth and also absorption of the nutrients from the soil. The first mechanism, that is nutrient solubilization. We know that some of the nutrients available in the soil, these are converts unavailable nutrients into absorbable forms. That means insoluble nutrients are modified into or converts into soluble forms, which are available in the rhizosphere. And phytohormones production. We know that phytohormones like auxin, gibberellin, cytokines, these are the phytohormones which are important for growth and development of the plants. And biological nitrogen fixation, using of the microorganisms, fixation of the nitrogen from atmosphere by bacteria, cyanobacteria, other microorganisms for fixation of the nitrogen from atmosphere. And biocontrol pathogens. Some of the pathogens are harmful microorganisms which are impact on plant growth and development and also the crop improvement. So these microorganisms suppresses the harmful pathogens for the protecting the usable and cultivated crop plants. Then induces systematic resistance, ISR, enhance plant defense against diseases. Some of the diseases caused the malnutrition and also the other developmental aspects, developmental problems we are observed in the plant growth and development. In that way, the microorganisms like PGPR, which acts to enhance plant defense against pathogens or diseases, some of the other plant diseases. Then important PGPR strains and their roles. What are the strains are useful for the improvement of the crops and also disease resistance. And what are the roles of this PGPR? Number one, rhizobium. It is a well-known bacteria for fixation of the nitrogen from atmosphere. It is a symbiotically fixation of the nitrogen from atmosphere in all leguminous crop plants, which induces the root nodules in all leguminous crop plants. And azospirillum, it is another type of bacteria which is also enhance the root growth along with the nitrogen uptake from the soil. Then Pseudomonas species. Pseudomonas also bacteria which produces antibiotics and also promotes plant immunity from the other pathogens and also other diseases. Then Bacillus species. The Bacillus bacteria act as a biocontrol agents against soil pathogens. We know that Bacillus thuringiensis is also a biocontrol agent. It is a biopesticide. We know that it is a BT cotton. Best example, BT cotton, very well-known Bacillus species. And then Trichoderma species. It is a fungal organism which improves root colonization and suppresses fungal diseases. So Trichoderma is a fungal member. It suppresses the fungal pathogens to develop the plants and also control the diseases from different fungal members or fungal pathogens. Then applications of PGPR in agriculture. So plant growth promoting rhizobia, which is helpful in agriculture crop plants by used these biofertilizers to improve crop productivity, applied as a seed coating for better germination and root development. We know that seed treatment is a better practices to germination of the seeds. The seed percentage, germination percentage also increased due to the seed treatment of the PGPR. And integrated pest management system, IPM. We know that integrated pest management strategies, which is helpful for the reducing the cost effects from the low marginal or marginal farmers for protecting their crop plants, and help in phytoremediation of contaminated soils. We know that so many soils are contaminated with some of the other what we can say that elements or minerals like thorium, selenium. So this type of heavy metals also impact on plant growth and development in agricultural crop plants. And commercial formulation available for various crops. This PGPR also commercially available in the shops. And PGPR also plays a vital role in sustainable agriculture. Sustainable agriculture is a very predominant term in present scenario. Why? Because farmers, they are knowingly or unknowingly, they are spraying a high amount of the pesticides and insecticides, some other chemicals that is harmful to the health of the human being and also the other living organisms. And this PGPR also plays the promoting plant health and reducing chemical inputs. The second, we can say that the green revolution is also the best example for the how importance of the chemical fertilizers and also chemicals, the plenty and the high amount and the highest quantity of the chemicals are used for improvement of the and increase the quantity and also the yield at the time in 1965-66 at the time. But nowadays, so the trends have changed. Why? Because so many factors are influencing to reduces the health. So that's why the PGP are also very important for the agricultural crop plants, increasing the yield also. Then some of the mycorrhizal fungal members, I am showing this table, different type of the mycorrhizal fungal members and what is the functions, what are the different functions of this mycorrhiza and the sustainable agriculture, how they are impact on sustainable agriculture and crop improvement. So first one, arbuscular mycorrhiza. We know that mycorrhiza are classified into the ectomycorrhiza, endomycorrhiza. So endomycorrhiza is the best example of arbuscular worm fungi. Worm fungi is the best example for the endomycorrhiza, that is the arbuscular mycorrhizal fungi. Then ectomycorrhiza, it is a formation of the sheets on the surface of the root. It is the substitute, it is the replacement of the root hairs on the surface of the rhizosphere or on the root. And ericoid mycorrhiza, ERM, which forms an association with plants in acidic and nutrient poor soils. Some of the soils, very high amount of the acidic conditions, and some of the soils, they are in alkaline nature. We know that. So in these conditions, ericoid mycorrhiza helpful the plants in acidic and nutrient poor soils. And orchid mycorrhiza. Orchid mycorrhiza is essential for seed germination and nutrient exchange in orchids. The examples of the arbuscular mycorrhiza. Am I audible, my dear friends? Okay. So arbuscular mycorrhizal fungi examples are the glomas. It is the best examples of the worm fungi. And rhizophagus, aculospora. These are the three important fungal members, examples for the worm fungi or arbuscular mycorrhizal fungi. And the Pisolothus and Lacaria and Boliotus, these are the best example for the ectomycorrhizal fungi. Ectomycorrhizal fungi. Then ericoid mycorrhizal fungi examples are Oidodendron and Hymenocyaphas. These are the examples for the ERM fungi. And last one, orchid mycorrhiza fungi examples are Rhizoctinia and Ceratobasidium. These are the examples. And what are the uses for the agriculture and crop improvement? Arbuscular mycorrhizal fungi enhances the nutrients uptake like phosphorus, zinc, and also improve the drought tolerance. Some of the stress conditions we are observed, drought tolerance, and also promotes plant growth and enhance disease resistance. These are the arbuscular mycorrhizal fungi significance. And ectomycorrhiza significances are helps in tree growth, enhances nitrogen and phosphorus absorption, and improve forest restoration and timber production. The ectomycorrhiza better suitable for the development of the forest and also the forest species. And ericoid mycorrhiza enhances the nutrients uptake in acid soils. Acidic condition soils are best suitable mycorrhiza, that is ericoid mycorrhiza, and also useful in horticultural crops like blueberries and cranberries. Nowadays, horticulture is a very, very important the emerging area for the commercial aspects, horticulture technology, gardening technology, lands, gardening. Okay. So these are in this field, this ericoid mycorrhiza is best suitable for improvement of the crop plants. And orchid mycorrhiza, it is the best suitable for the improvement of the orchids and propagation of the orchids, and improve seedlings and survival rate of the orchid cells by promoting this orchid mycorrhiza. Then we have already discussed these benefits of the mycorrhizal fungi. Now, nitrogen fixing bacteria. We know that nitrogen is the most important nutrients for all plants. Nitrogen is highest amount of the gaseous substances which is available in the atmosphere, but it is a molecular state of the nitrogen which is not available or unavailable to the higher plants. Due to that reason, the atmospheric nitrogen is conversion into usable forms of the nitrogen. That is ammonia, nitrates, and nitrates. These are the usable forms of the nitrogen. But this nitrogen is conversion by the microorganisms like bacteria, cyanobacteria. So these bacteria and cyanobacteria members they are symbiotically or asymbiotically. They are fixing the nitrogen from the atmosphere due to presence of the nitrogenase enzyme or dinitrogenase enzyme. This nitrogenase enzyme controlled by nitrogen fixation genes or NIF genes. The best examples of these symbiotic bacteria are the cyanobacteria or the symbiotic bacteria Rhizobium leguminosarum, which induces the root nodules in legume plants, and free living bacteria, that is the